This is a production of Cornell University.
Yeah, thank you. I'm Maddie Oravec. I am an assistant professor in the horticulture section, and my lab works on grape breeding, genetics, and genomics. New York is the third highest grape producing state, and the wine and grape industry has an economic impact near $7 billion. So it's quite a big industry, and it's a fairly diverse industry too across the state. I inherited and am stewarding the Cornell grape breeding program, which is nearly 140 years old. It's nearly as old as the experiment station where we're housed, the New York State Agriculture Experiment Station, now Cornell Agritech in Geneva. And while the industry is primarily wine, and we are focused primarily on wine, there is a large Concord industry, juice industry in New York as well, and we also work on some table grapes in our program as well. Oop, there we go. As with most industries right now, we're faced with a lot of changes with climate change, changes in kind of climate pressures and disease pressures, increasing production risks and disease pressures. And we're also facing regulatory changes and fungicide resistance that impact spray efficacy and availability. Wine grape production, or grape production in general, takes a lot of disease management. And so, on a good year, they might be spraying every 12 days, sometimes more likely weekly. So disease management is a major focus for grape production, and for us, disease resistance, therefore, is really important in our breeding program. And of course, the societal, economic, and environmental pressures to improve sustainability of production, both kind of for the growers' profitability, as well as for the environmental sustainability. And so one of the bread and butter of the grape industry is Vitis vinifera. This is kind of the ones you might know: Merlot, Cab Sauv, Riesling, and these are known for great quality, but they're quite cold. They're majorly cold sensitive and very disease susceptible. And so while we use this as an incredible base for our program, it has a wide diversity of colors and sizes and shapes and all of that. We also have to rely on a lot of wild species, and we are fortunate that in Vitis, we are breeding actually across species. So these are interspecific hybrids, not necessarily the direct result of a cross between a wild and a vinifera, but kind of integrating these wild species into this vinifera background to get the benefits of cold hardiness, disease resistance, kind of these resilience traits. We have over a dozen or dozens of wild species that are actually native to the US, but then also other species around the world. And we can draw on this incredible diversity and these naturally occurring selected traits for disease resistance. But as we probably recognize from just going around, and I don't know, maybe everyone else also tastes the grapes on the trails when you're walking around, they don't have that same quality that we expect for production. They don't have the same cluster size, productivity, cultivatedness. So in our program, we're utilizing these wild resources to bring in the traits we want: the disease resistance, the climate resilience, and using Vitis vinifera for those quality traits. And so in our grape breeding program, our mission is to explore and improve grapevine resilience and quality through phenomic, genomic, and stakeholder engaged approaches. And so I'm going to just highlight today in this quick flash talk format kind of some of the different approaches we're using to address this. So novel on high throughput phenomic approaches, we're using various technologies. Cornell Agritech, we have a really rich body of people working on these kind of new technologies for high throughput phenomic approaches. And so we're utilizing these tools in the breeding context. And so one example is this Blackbird. This is basically a 3D printer that's been kind of hijacked to take images of leaf discs over time. And so this is a way that we can have a controlled environment for testing disease resistance primarily in this case, but it can measure kind of anything that you can see on a leaf disc, where you can do a controlled inoculation and image over time to get kind of a quantitative measure of the difference in susceptibility or resistance. We also are utilizing this robot that drives through the vineyards and takes pictures of the vines. And so we're using previously described models for powdery mildew and downy mildew, which are two of the most important fungal diseases in grapevine production. But we're also, this video shows one of my graduate students, Aiden, who's working on training this on phenology traits, but we're also interested in other diseases and growth measures and anything else we can kind of pick up from these images. And then we're also using some hyperspectral approaches. So these hyperspectral images give us kind of these measures of reflectance across a wide spectra. And so this allows us to pick up on things that are maybe not visible to the classic imaging approaches. And so we're doing this in two formats. One is in this hyperbird. So it's basically a black bird, but with a hyperspectral camera. And then also kind of a scanner, a large, more macro scale, where we're looking at berry. And one of my new master students, Matthew, is working on this in terms of ripening traits and berries in our mapping population. And so this is a picture from last year of some of those berries on a tray being imaged by this hyperspectral scanner. In terms of the genomic approaches or genetic approaches, a really important part of our breeding program is using marker-assisted selection. There's been a huge library of markers that have been developed through the Vitis Gen projects that are SCRI grants. We are now in the third iteration, Vitis Gen 3, and now we're part of another SCRI, Vitis by Muscadine, where we're trying to kind of bring traits from Vitis and Muscadine and trading across these two markets. And so by improving our phenomics and also our genomic characterization, we can get these DNA fingerprints, where now instead of having to do this phenomic screening, we can use these DNA markers to detect the trait we're looking for and do the selection early in the selection pipeline. And it works. One of the most important loci that we work with is run 1. This is actually like a combined powdery mildew and downy mildew, run 1 RPV1 locus. And with it, our foliage is quite clean year to year, low downy and powdery pressure. And this is in a low to no spray vineyard setting. And without, you can see a vine will be just completely decimated by powdery and downy mildew. So this is really important to us, and continuing to develop these markers and use them in the program is essential. But it's not so simple. Resistant isn't always resistant. We know that the RENS3 locus, which is another marker for powdery mildew resistance, doesn't really work in our region. We are actually in the center of origin for powdery and downy mildew. And so we have a great diversity of pathogens that are really good at overcoming some of our resistance genes. And so one of the things that we also have to work on in our program is stacking these resistance genes so that we hopefully have different methods of kind of barriers for these pathogens to overcome our resistance. But also, this is why testing across environments is so important. We know REN3 is in a lot of the older French American hybrids, so it's been growing in this region for a long time, and the pathogens have had just a lot of opportunity to overcome this resistance. And so we have to think about stakeholders across the spectrum. So we think about kind of the nurseries growing and propagating these plants, the growers, of course, producing them as a major stakeholder, but also the winemakers, and then, of course, consumers. And so we have different traits. These are just a few examples across the spectrum that we have to think about in our program. Vigor and vine establishment, productivity and yield, as well as this disease resistance. For the winemaker, uniformity, winemaking quality, whether there's this expression of genotype by environment, or as we like to call terroir in grape, and of course for the consumer, the color, flavor, aroma, mouthfeel. And so we have a big list of traits that we have to evaluate to find these super grapes that have potential in the industry. And so as part of this, we also recruit commercial partners and commercial input. One project I'm working with Katie Gold and her graduate student Nico with a big block that they have of one of our advanced selections on MYO6. They're doing some management trials, but at the same time, we're working with some commercial partners to produce commercial wines, because in our own program, we're producing research wines, but there is some challenge. There is some negatives to that. And we're also hosting industry events for tasting these research wines. So now I'll just take you on a quick photo tour of kind of the breeding process. We start in the fall or spring with emasculating flowers. This is usually a fast and furious couple weeks in June. And if you think about a grape cluster, every single one of those grapes is originally a flower. And for every single one of those flowers, we have to take the male plant parts off the anthers and leave behind that undamaged receptive stigma to make our crosses. And so this is obviously a huge bottleneck and a big push and effort in our program every June. We then bag those, make that controlled cross, and are able to harvest those fruit and seed, plant those into the greenhouse. This is where we can collect tissue to get DNA to do that early selection of those markers. The ones that make it through that selection make it out to our no-spray nursery, where we can do the first round of phenotypic evaluation of disease resistance. The ones that make it from there get out to a permanent vineyard, and if we propagate them from there to 6 vine plantings, we can get enough fruit to harvest. We make wines, and we're working with the Cornell Craft Beverage Institute, and then we use a in-house training panel to do our wine evaluations. So it's a whirlwind and it's a lot of fun. And yeah, that was just kind of a taster of some of what we do. Thank you.
So I wanted to do 2 things with my lightning time today. One was to answer this question, which I've thought about across all of my time at Cornell. Why have a public sector field corn breeder at Cornell? People ask me that question within the university. People outside the university say, don't you want to be in Iowa? And the answer to that is no. So that I want to talk a little bit about that. And then I want to give you an unabashed teaser for the seminar I intend to give in the spring in plant breeding and genetics that will really recap some of the research I've done over the course of my career. I'm at the opposite end of the spectrum from Maddie. So now you're looking at the person who's already on phased retirement. So why a public sector field corn breeder at Cornell? This is just a map of growing degree days in July. You can see New York has a lot fewer than the Corn Belt. That's an advantage they have. They also have soil. We have rocks and clay. They also have nice weather, like today in the fall. We tend to have clouds and mist. So there's a lot of reasons why corn grows really well. Cornell doesn't grow really well. Corn grows really well in the US Corn Belt. It also is really important, however, here in New York. So field corn production in New York is on about 1 million acres, about half for grain, half for silage. That is largely in support of the dairy industry, which is one of New York's biggest agricultural enterprises. I've listed the grain and silage yield averages for 2024 there. You can see from the rank, we're like middle of the pack in terms of our yield potential and our silage yield potential. Both of those have gone up a lot over time. However, we're not going to be up there with Iowa anytime real soon. That said, just to make it clear that this is an important crop, this is all of New York crop acreage in the 2024 growing season and how much of it was planted to what. And you can see very clearly the crops that support dairy: alfalfa, hay and haylage, non-alfalfa hay and haylage, that big orange part, and field corn and soybeans, and then a whole lot of other stuff that we grow a lot of, many different crops. But field corn is the single largest row crop grown in the state. So there's a good reason for having a public sector field corn breeder at Cornell. Many years ago, people in the Crop Science Society started to look at the capabilities for breeding across private and public sectors. So they did this study of scientist years in corn breeding back in 1996. That's like 30 years ago. I would like to say that was early in my Cornell career, but it was sadly not that early in my Cornell career. At that time, there were over 500 FTEs, full-time equivalents, doing corn breeding in the private sector, and a total of 35 in the public sector, of which only 3 focused on cultivar development. You're looking at one of them. Since that time, they updated that study in 2015, 10 years ago. Public sector field corn breeders were down and private sector were up. And I have to say that my appointment here is research extension. So I'm at that very applied end of the spectrum, or translational end, whichever way you care to talk about it. But I'm one of those people who actually tries to do development of varieties in the public sector. So that it makes me look a little like a dinosaur, right? Over this same time frame, 1996 to about the, I think it's to 20, or sometime in the 2000s, there was a study done of seed industry consolidation. 2022 is the latest version here. So this is kind of like a pedigree of the seed industry. And you can see that there are really, if you could see all the lines there are really three huge groups. The top is Bayer Monsanto. The left bottom there is ChemChina, which used to be Syngenta. The right is Corteva, which includes Dow, DuPont, Pioneer, that whole group. Huge consolidation in the seed industry over this time period. Remember, this relates back to those 500 breeders. They now work for many fewer companies, so lots of fewer avenues for different genetics to come through that pipeline. As a consequence, this picture shows just the market share of seed sales for corn, soybean, and cotton. I'll focus on the corn one, but you can see that there are two companies that account for over 70% of the corn seed sold in this country, and the next biggest one is a very small share. So not a lot of diversity feeding into that pipeline. So that, I think, is a good reason for having a public sector program in an area of the country where there is not a single private sector breeder operating. That would be here. So what does a public sector corn breeder in New York do with a small program that does not have many resources compared to a huge number of breeders in the private sector with very well-funded large programs? I think there are a few opportunities. The goal for the private sector obviously has to be profit, and they do that largely through increasing yield potential, so they have outstanding hybrids, and making sure those hybrids have a large area of adaptation so they can sell lots of seed of them. That opens some little windows for the public sector. One of them, which my program has engaged in, is variety testing, making sure that those things bred elsewhere really do well here in the Northeast and in New York, comparing across companies, which companies' own variety testing efforts never do. Variety development for what I'll call production niches. Basic germplasm improvement, the longer term stuff that the private sector often doesn't engage with, and of course, plant breeding research. I'm going to talk today for a moment about just a couple of those, not all of those. So the focus of my breeding program has really been what I call niche environments. New York, it's a niche in the sense that it is not the Corn Belt. So it's one that's kind of left outside of the major focus of the private sector. I also have done a lot of work, which I won't talk about today, with smallholder farmers in the tropics for whom very little directed breeding work has been done. That has been largely through collaborations and graduate students. We have done a fair amount of commercial silage hybrid testing. Lots of our corn here is grown as silage. Silage testing is expensive. The private sector doesn't like to do any more of it than they have to, but it's important to our growers here in New York to be able to answer the question, which varieties across the spectrum of companies are good for silage production here in the Northeast. I have done some hybrid development and testing. I had a couple hybrids that were marketed for a while, and then biotic and abiotic stress tolerance. So these are kind of the areas where I focused. So the variety development for niche environments, it's good to ask the question, what's not being fully addressed by the private sector, because they are addressing a lot. Short season environments, back to that picture I showed you, is one of those things. We have cool, wet springs. We have rainy, cloudy falls. The Corn Belt does not have much of either of those. And we have a shorter growing season. So things that fit those short season environments. Another area that I've focused on is organic production systems. And I'm happy to say I've had some commercially marketed hybrids for the organic sector. In terms of germplasm improvement, this is a plant path seminar, so I figured I had to show you some diseased plants. My position, the one I occupy now, was first established in 1970. I did not occupy it at that time. And it was established, what happened in 1970? I know Gillian knows this, the Southern Corn Leaf Blight epidemic. It was established precisely to address disease and insect resistance in corn. So that focus has continued to this day. I do a fair amount of work on selecting for resistance to European corn borer. Yes, we have BT. On the other hand, every farmer who uses it needs to also plant a refuge, and there are many farmers who do not choose to use BT for whom insect resistance native to the plant is useful. In terms of diseases, some of the major focus areas we've had are gray leaf spot, northern leaf blight, and anthracnose stalk rot. So the top picture there is shows one of our gray leaf spot resistant hybrids next to one that is not just old, it's dead due to gray leaf spot. And on the bottom, you have one of our anthracnose resistant hybrids inoculated down here, as you can see, versus a susceptible check. So we've made some good progress. That's the teaser. I'll be doing a seminar in the spring to talk much more in depth about my research program, so keep an eye out for that. Lastly, none of this would have been possible without lots of fantastic people over the years from many different places, and these are just a few of the more recent ones. Some of my field techs, Sherry, Keith, and Dan, my most recent graduate students, all of whom have graduated. And so with that, I will just say thank you.
Everyone, thank you so much for this opportunity to briefly talk about the research for my group. So I've been here for a little bit over a year now, and this is what we do in our group. So I'm assistant professor with the soil and crop sciences, and what we do is the water. So we look at the plant water use with remote sensing at different scales, from the field to the global scale. So water is essential for lives, and water is important for agriculture. We all know this, right? Farmers need to make a lot of decisions overall, and water is one of these key decisions. So they either related with like how much water they want to irrigate, when to turn on the irrigation, or this related, like water related on-farm practices, like they look at the weather forecast to see, okay, what I need to do for the next week, whether I want to plant it or not, whether I want to harvest or not. So water is so critical for agriculture, and how we can measure the plant water use. Traditionally, this is hard, and even now it's still hard. So my group uses remote sensing algorithms to estimate this water use. So the water use is not directly measured from the chamber or the sap flow. It's looked from the satellite, from the drones, that we look at the evapotranspiration. So how much water has been used? You can look at this, how much water can be transferred back from the soil into the atmosphere, which is this evapotranspiration process. And this process is invisible, which makes it harder to measure. It includes the evaporation, that is water returned back to atmosphere from different surfaces, and also the transpiration, which is a part of that the plant uses water, draw the water from the soil, return back to atmosphere. So combining together is this evapotranspiration. So evapotranspiration is really not just a water process. It's not just basically, the water from soil into the plant, return to atmosphere, then fall back as precipitation. Well, that's part of that. Another part is also energy flux. Why is it energy flux? Like you can think about in the hot summer, when you sweat, you feel cooler, right? So it's the same for vegetation or plant to use water. When they use enough amount of water, return back to atmosphere, is also the cooling effect. You will see a lower temperature for those well-irrigated plants comparing with the stressed. So also with the stress signal, we can use the evapotranspiration as approximate index for the stress and also disease. So why this is, we can look at that because evapotranspiration, it uses stomata, which is the same path as the photosynthesis. So when the plant gets stressed, the first thing is not turning the leaves to yellow, which like a lot of vegetation indexes use. It's not that quick. But the first response is close the stomata, which will also decrease the ET. If we can capture that, we can also detect the stress. So here you can see the evapotranspiration is not just water. It connects the different scales from the stomata level to a single plant, to a field, and also to the global. It impacts the entire atmosphere, the energy fluxes. So my group uses this multi-source remote sensing data together to improve the evapotranspiration estimation at both temporal scale and also spatial scale. So which means we can see the field clear and more often. So here you can see we use a bunch of data sources from different satellites, not just from the US and also from European. We use the drones that we fly in the field. We also use the sensors that we built into the field together to look at the evapotranspiration. And all these data, they are different. And they have their uniqueness, they have their advantages. What we want to do is we want to get all these advantages together to help us estimate a field level daily evapotranspiration. A field level here in remote sensing means 30 by 30 meter spatial resolution. So basically like a little bit larger than this room. That's a pixel. So we can really see the signal from all the remote sensings for at that level. Okay, so for the model we work on, it's actually requires a lot of computational resources because that's a very high resolution. And even just to store that data, it requires a lot of spaces. So like when we want to expand to larger area, our local server just cannot do it. So how we can solve this bottleneck actually gets solved by the cloud computing. So instead of making our local server larger, stronger, bigger, what we do is we use the cloud computing. We move our algorithms into the cloud and do all the computations in the cloud. And also we save the data in the cloud, which actually make it easier to communicate our results with end user. So this effort is done through the OpenET project. I've been working on this project for many years. Before I joined Cornell, when I was a postdoc, I already started work on this. So this is a really collaborative effort from multiple institutions, agencies. We call it OpenET because we want to, this ET data is open to everyone. So I need to say we are very successful on this now. So we already have our official data launched. The structure here is like I just mentioned, instead of downloading data, store data, we upload what we need into the cloud. So what the cloud platform we choose is Google Earth Engine. The reason we choose it is because they already have most of the data we need. So we just need to upload like tiny amount of data there and basically just implement our algorithm there. So now we are operationally produce 30-meter daily evapotranspiration data on the Google Earth Engine, and this data can be found on this website, openetdata.org. We formed a nonprofit organization with this, so you can find all this data here. And also for a more advanced user, you can also try our API platform that you can directly download a bunch of data in a short time. And also we are developing a farms platform. The name is Farms. So the farmers can upload the field they are interested and get a report every month or every 2 months based on how they want to look at it. So this is a screenshot of the OpenET website. Basically, we don't really cover the entire CONUS yet, but it's very close. We are putting a lot of effort to expand to cover the entire mainland of United States. So you will expect to see this. And this is just a short animation to show you that you can see the water use from each of the single field. And you can look at the water use history for each of this field. To use this data, my group use this for vineyard irrigation scheduling in California, which is a project supported by NASA collaboration with USDA and also ENG Gallo. We also look at the the soybean and corn water use patterns over the Mississippi alluvial plain, which is also a major agriculture area. So a more related and recent initiative of the effort is we are moving our study area to the Geneva site. So this is a collaboration with Katie Gold and Yu Jiang. We built 2 flux towers and soil and radiation towers in one of Katie's vineyard. So we are monitoring the carbon and water flux 24 hours, 7 days. Since the starting of this growing season. So I'm super excited to start to analyze the data after this growing season. And not just the ground-based instruments, we also look at the satellite overpass to get the satellite data. We have Nico from Katie's group that he's flying the drone, that he's also provide the UAV images to us that we can also use that to calculate the water use at a fine scale. So it's really a multi-scale, multi-temporal resolution effort together. So this is just an initiative, so stay tuned for more results for this. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.